So this is an example of how to explore a chemical-induced transcriptional signature in NextBio. So I am going to use Fenofibrate to search the database. This is a drug that's used to lower blood lipids. And once I search it, this quick view will show up, and then I will go to curated studies. Hopefully this will show up quickly. There we go. And I am going to search for liver gene expression in rats. So first I'm going to go organism. I'm going to hit apply. And then I'm going to go to liver, which most of them will be. Apply filter. And just click apply filter here. And then I'm going to go to the study called Genomics Assisted Toxicity Evaluation System Study Rat Liver Repeat Dose. Okay. So this is a study, this is from what's called the TG Gates dataset. And what I'm going to do here is simply pick the first one that shows up here, liver from Sprague-Dawley rats, 24 hours after last dose. There were 14 days of exposure, so 14 repeated doses, and it was at 1,000 milligrams per kilogram, and there were 5,119 features changed. So this is the gene list that we're going to work with here. So I click that little window next to the gene list, and what I get here is the list of genes that are differentially expressed. Okay. Now that we have our list, we can do a number of things. The first thing that we're going to do is actually look at the other curated studies that are similar to it, and we're going to look at a distinct data set called drug matrix, and the way we're going to find that once I get it in here. We will go to organisms, and we'll first filter for rat, and then we'll go to keyword, and we're going to type in, so SEBS is the database that holds the drug matrix data, and we'll hit apply. So now we'll search for rat and SEBS, and what comes up is the drug matrix in vivo toxicogenomic study in rat liver Affymetrix. And then we're going to go in here and we're going to take a look at what treatments have highly similar responses to our treatment list, basically our list of genes. Basically it's a correlation analysis that's run, which gene lists correlate best with ours. And so Fenofibrate is a lipid lowering agent that activates PPAR alpha. And the first chemical that shows up here is a lipid-lowering agent that's not on the market because it ended up causing cancer, but is also a very potent PPAR alpha activator. And the phenopin is also a very potent one. Then, of course, there's Fenofibrate, bezafibrate. Again, so what you're seeing here, what this illustrates is that chemicals with similar pharmacological properties produce very similar responses in gene expression. And so what this is liver. Gene expression from liver treated with pyrenixic acid. This is an in vivo study at this dose level by oral gavage for 3 days compared to vehicle. And so if you do this comparison, you can see that here's our Fenofibrate data set, 14 days, 1,000 mg per kg, versus the pyrenixic acid. And you can see there's a very strong overlap, and there's a high degree of concordance up, up, down, down. And so if you look at the overlapping genes, ACOT1 is always the most strongly expressed gene when you give high doses in rat, particularly when you're measuring in liver. And so ACOT1 in our data set is 729-fold changed, and in the pyrenixic acid data set is 696-fold change. So that's how you identify the top most similar chemical treatments in another data set that used rat liver. The next thing we're going to want to look at is the body atlas. So we're just going to come back up here. So we've done all this filtering, and Doesn't really matter. We're still working. We know we're working with this treatment here, this 14-day fenofibric acid gene list, and we're going to go to Body Atlas. And first thing you see here on Body Atlas is the list of organ systems. So there's different things you can look at. You can look at tissues, cell types, cell lines, stem cells. The only one I want you to look at here is the tissue one, and we're going to look at Homo sapien for now. There is no rat. But what's interesting about this is green indicates an inverse relationship. So genes that tend to be disproportionately up in liver in normal liver are actually downregulated, which is indicative of a dedifferentiation. And if you open this up here, you can see that there's an inverse relationship between the chemicals, the effect of the chemical on rat liver, and the genes that are regulated, or the genes that are expressed in liver on a relative distribution to other tissues. So there's an inversion of that. So basically what's happening is when you treat with fenofibrate at high doses, you're getting a dedifferentiation, most likely due to the damage. So next we're going to look at the disease atlas. Most likely what's going to show up here is basically we're going to take our list of genes now and throw it into their curated disease atlas, and basically these are lists of differentially expressed genes from tissue, and not surprisingly what you have is injury of liver coming up highly positively correlated as the most strongest effect based upon their scoring system here. And you can click on this and open it up, and you'll see that it's related to probably to damage that was induced in a rat model. Next, we're going to look at the pharmacoatlas, and it'll load here in a second. Now we're going to take our list and look at how our list of differentially expressed genes, and look how it relates to gene sets that have been curated associated with certain pharmacological perturbations. And not surprisingly, this is an incorrect characterization of the database. They're not really anti-metabolites. They are PPAR alpha activators, but the top scoring one, not surprisingly, are all that's a PPAR alpha activator. So is that. I'm not quite sure what that one is. That's a PPAR alpha activator, and that's also a PPAR alpha activator. So not surprisingly, the top scoring category are all loaded with PPAR alpha activators. So now, finally, we're going to take our gene list here, and we're going to look at the knockout atlas. And the knockout atlas, basically, we're now comparing our differentially expressed gene list to lists of genes that manifest as a byproduct of knocking out a gene. And voila, not surprisingly, when you knock out PPAR alpha, you actually get a negative correlation. So you could open this up if you wanted to, and what you'll see is that the genes that are typically upregulated with our treatment are downregulated when you knock out PPAR alpha, which indicates that PPAR alpha is probably the receptor mediating the effect. Next, we're going to have a look at the pathway enrichment. With PPAR alpha activators in the liver regulates lipid levels in the blood, and that's what fenofibrate is targeted towards, affecting metabolism of lipids in the liver. So some of your top scoring pathways and gene sets should be associated with metabolism in the liver, particularly fatty acid metabolism. And there you go, not surprisingly, the fatty acid metabolic process is one of the top scoring pathways. And if you actually go up here and just show canonical pathways, what most likely will occur, things involved in metabolism of lipids and lipid proteins. So you can see here there's a number of gene sets in the top scoring sets of genes, all again directionally upregulated here, that are associated with our gene list from fenofibric acid or fenofibrate treatment for 14 days in rat liver. The final thing that I would want to do here real quick is actually to come up with a signature. So what we're going to do is we're going to have this loaded here. We're going to go back to curated studies, and then what we're going to do is we're actually going to go into this genomics assisted toxicity evaluation rat liver repeat doses. And what we're going to do is we're going to come up with what I call an open signature, right? So what we're going to do is we're going to press this little green button here with the funnel on it. And what that's going to do is load it up here in the meta-analysis. So basically you're going to mine for a signature. And so what we're going to do is we're going to take all of the repeat dose from this genomics-assisted toxicity evaluation system study with rat liver repeat doses, and we're going to find all the fenofibrate treatments. So we're going to go here to keyword and go fenofibrate. Then what we're going to do now is we're going to add every single one of these treatments to the meta-analysis tool. So now what we have is a collection of gene lists that we can now mine. So we're going to click on the meta-analysis here, and I'm just going to leave the settings for the filters to be up or down. And so basically we're going to go and look for genes that are consistently affected by the fenofibrate treatment over multiple different dose levels and multiple different durations, 14 days, 28 days, 7 days. You can see this here in all the different doses, 1,000, 100, 1,000, 10s down here. And so we'll go here to the gene result. And what you'll find is, not surprisingly, ACOT1 is very strongly affected in all treatments, and a number of these other genes that are associated with lipid metabolism are also this ACOT2, peroxisome biogenesis, acyl-CoA thioesterase. These are all related to VNN1, all related to lipid metabolism. And so you now have a signature, so the question is, of course, what does that signature represent? And so what you can do is jump over here to the biogroup results, and it tells you what sort of pathways and gene sets are disproportionately affected by your basically your signature, what your signature is enriched for. And so it'll quick show you this here. Hopefully, it's taking a moment here. And what you can do is you can go to canonical pathways to look at what the curated pathways look like. And it should be, if this worked correctly, it should actually show up with something related to fatty acid metabolism. There it is, genes involved in metabolism of lipids and lipoproteins, fatty acid metabolism. So that's how you make an open signature in the meta-analysis tool, right? Well, we have all positive treatments, we have no negative treatments, we have no anti-fenofibric acid treatments. That's why I call it an open signature. And you've gone here and you used the meta-analysis, you did the setup, you then basically, you said genes can be up or down regulated, you just have to be consistent across the treatments. You went to the gene result, identified those genes, ACOT1 being one of them, and then of course there is the biogroup results. So that's it. That's how you evaluate fenofibric acid and also generate a signature for fenofibric acid treatment or PPAR alpha activation in rat liver.